Thank you to all who have taken time out of their busy schedules to attend this presentation on improving the quality of allergy and respiratory clinical research using induced sputum biomarkers. The presentation will be split between my colleague, Professor Neil Alexis from University of North Carolina and I, and we hope you find the next 45 minutes to an hour very useful. Next slide, please. And again. Thank you. As we all know, increasing R&D expenditures do not translate into increasing drug approvals. On the left vertical axis, you see the number of new drug approvals, and on the right vertical, you see expenditure in billions of dollars, with years starting from 1963 on the x-axis. It is a well-known fact that while the number of new drug approvals seem to undulate around a baseline, expenditures have skyrocketed. Next slide, please. This graphic contains a lot of information. Let's just focus first on the number of research programs going from left to right. For every approved drug or successful program, we will have 1.6 in phase 3, 4.6 in phase 2, 8.6 in phase 1, 12.4 in lead development, and 24.3 in target validation and lead selection programs. If you look at the dollar amounts of capitalized costs on top, you realize that phase 1 to 3 are approximately equally expensive, and that lead optimization before phase 1, with $150 million for all 12.4 programs, is significantly less expensive. This will be the place where extra expenditure for lead optimization would have the largest downstream savings. Of course, there's only so much optimizing you can do at this stage. Several initiatives pursuing precision medicine and personalized medicine approaches are exploiting these areas, notably in the world of genomics. But through better study planning and design and targeted approaches to biomarker development, we could change that too. Next slide, please. In this slide, we see asthma and rhinitis studies in June 2014, and we see how many studies are occurring in phase 1, 2, and 3. But it's noted also, with the increasing use of generics or development of generics, with the reduction in blockbusters from 2014 to projected 2025, coupled with an increase in more personalized or phenotype-targeted therapy in biologics and biosimilars. Next slide, please. If we take asthma as an example, a factor analysis has showed that certain biomarkers, i.e. lung function, airway hyperresponsiveness, and airway inflammation, comprising distinct groups of disease determinants which are not necessarily interrelated but rather complementary, provides complementary information on the disease phenotype. Hence, in proof of concept studies of asthma, all these aspects or factors should be evaluated, and ideally perhaps in clinical monitoring of asthma as well. All biomarker indices provide distinct and complementary information on a certain asthma phenotype, but also addressing individual phenotype response to therapy. So like the classical concept that say lung function mirrors changes in airway inflammation, in clinical trials different aspects of the disease like asthma should be assessed. And there are at least 3 groups of determinants of asthma. 1, clinical and lung function obstructive parameters. 2, airway inflammatory markers. And 3, markers of airway hyperresponsiveness. Based on a factor analysis study published in JACI a few years ago, it appeared that the clinical and lung function parameters of airway hyperresponsiveness and airway inflammation are all independent markers of asthma, providing complementary information that may lead to differentiate among the distinct asthma phenotypes and help to monitor the response of new anti-inflammatory therapy. Next slide, please. Adding more subdivisions, for example, clinical, physiological, triggers and inflammatory components, many different phenotypes could be identified. In fact, combinations of many biomarkers within one individual may finally define its asthma phenotype. And as you can see, there's an extensive overlap among the different phenotypes. So in view of the heterogeneity of asthma, several authors have investigated and published on different asthma phenotypes, and although not fully standardized, here is a potential proposal by Sally Wenzel for subdivision in asthma phenotypes, clinical or physiological, those who are sensitive to specific triggers, and inflammatory phenotypes. But of course, there is a lot of overlap amongst these phenotypes, and novel detection techniques in preferably non-invasive airway samplings should help to link the clinical and the physiological components to the underlying immunology to define an individual's asthma phenotype to enable that targeted therapy. Next slide, please. Thank you. So this is a busy slide, but one which I hope draws attention to the role of system or network biology. Identifying those who respond to certain stimuli, such as allergen, and as individuals produce selective increases in key biomarkers of interest, such as IL-5 and prostaglandin D2, as we see in this schema here, we're able to identify certain responders, those who have the potential to produce increased amounts of certain mediators and signals in relation to a certain stimulus. And of course, we don't necessarily need to use allergen as an example. We can use LPS or virus to look at how we can draw inferences for what we see in vitro and ex vivo to develop those strategies in clinical development in vivo, to look at those who might have a certain dose response and those who perhaps are predicted responders to that type of influence or stimulus or indeed that therapy. So we can use techniques to emphasize the target of interest in human, in vitro, or ex vivo to create the confidence in measuring your biomarker once in the clinic, in vivo, which is critical to developing sound, go, no-go decisions. Next slide, please.